Welcome to the module of hurdle technology. Hurdle technology is a technique to control and eliminate pathogens by combining two or more factors. These factors are called hurdles. The pathogens have to overcome these hurdles to survive. The right combination of the hurdles inactivates the pathogens and thereby makes the food products safe for consumption along with an extended shelf life. Hurdle technology is a technology where appropriate types of hurdles are selected and combined to provide microbial safety, stability, sensory, nutritional value and economic viability to products. Now let us move on to the objectives of hurdle technology. After going through this section, you will be able to explain the basics of hurdle technology and combine various hurdles to preserve different foods. Now let us move on to see the need for hurdle technology in the current scenario. The demand for fresh and natural products, the need for minimally processed products, trend of eating out and consume ready to eat foods, and finally it is the emergence of new routes for the growth of microorganisms. Now let us see what is a hurdle effect. The combination of various factors is called hurdles. The combination of various hurdles improve microbial stability, sensory property, nutritional quality and economic property. There will be a significant synergistic effect between hurdles. For example, gram-positive bacteria include some of the most important spoilage bacteria such as Clostridium, Bacillus and Listeria. A synergistic enhancement occurs if nisin is used against these bacteria in combination with antioxidants, organic acids or other antimicrobials. Now, combining antimicrobial hurdles is an intelligent way so that other hurdles can be reduced, yet the resulting food can have superior sensory qualities. Stability and safety of foods depends on various preservation factors which are called the hurdles. Depending on the type of product, the type and intensity of the hurdles will vary case by case. The effects produced by the combination of these hurdles is called the hurdle effect, which is used to preserve the stability and safety of foods. Now let us move on to the types of hurdles which are used for preserving foods. The combination of various preserving factors is called hurdles. There are different types of hurdles used in foods like high temperature, low temperature, water activity, acidity or pH, redox potential or oxidation reduction potential, and preservatives like nitrite, sorbate, sulfite and competitive microorganisms like lactobacillus species, specifically lactic acid bacteria like Lactobacillus acidophilus, Lactobacillus plantarum, Lactobacillus delbrueckii and many more. The concept of combining several factors is known as hurdle effect. Food preservation could be attained by different types of hurdles. The different types of hurdles used for food preservation are classified into three types: physical, physiochemical and microbial hurdles. These hurdles also influence the quality and safety of foods. Depending on the intensity of different types of hurdles used, their effect on food quality may be either positive or negative. The main objective of hurdles is to eliminate and inactivate or inhibit the growth of microorganisms present in food. Common salt or organic acids like citric acid, lactic acid and acetic acid can be used as hurdles to control the microbial load in foods. Natural antimicrobials, also called as biopreservatives like niacin, bacteriocins and essential oils can also be used as one of the hurdles. The emergence of hurdle technology is due to the following reasons as given below. First one, the demand of the consumer for healthy and nutritious foods. Secondly, the shift to ready to eat, ready to cook, ready to serve and convenience foods which requires less time and less processing. Third one, preference of the consumer for foods with less chemical preservatives. Foods contain hurdles and parameters like F value for high temperature processing, T value for low temperature processing, low water activity, acidity or pH and low redox potential and preservatives. There are few microorganisms present in food which can overcome a number of hurdles, but not all the organisms can overcome the different types of hurdles used, thus making the food stable and safe. Now let us move on to the types of hurdles with specific examples. There are three types of hurdles. One is physical, physiochemical, and the third one is microbial hurdle. Now let us see about the first hurdle, physical, and its specific examples. So under physical hurdle, there are different types of examples. For example, like high temperature sterilization, evaporation, pasteurization, extrusion, baking, frying and blanching can be included. In low temperature, freezing and then modified atmosphere and controlled atmosphere technology can be applied. Then comes ultra high pressure, ultrasonication, ultraviolet radiation, ionizing radiation, aseptic packaging, packaging films, electromagnetic energy, which includes microwave, radio frequency and pulsed electric field. Second type of hurdle is the physicochemical hurdle, which has examples like carbon dioxide, ethanol, lactic acid, lactoperoxidase, low pH, low redox potential or oxidation reduction potential, low water activity, organic acids, oxygen, ozone, phenols and phosphates, along with spices and herbs. And third type of hurdle is microbial hurdle, which has examples like antibiotics, biopreservatives like niacin and bacteriocins. And it finally includes competitive microflora, which includes your lactobacillus species like Lactobacillus acidophilus, Plantarum, Delbrueckii, Casei, Paracasei and many more. Most important hurdles for food preservation and their symbols. The first one is F, which indicates high temperature and the application is heating. Second symbol is T, which denotes low temperature and it is used for chilling or freezing. Third one is water activity. We have to use reduced water activity, which is obtained by drying or curing. Fourth one is pH. Its application is acid addition or formation of acids. Fifth one is EH symbol. It is the symbol of reduced redox potential or oxidation reduction potential. Application is this. It is nothing but removal of oxygen or addition of oxygen as required by making use of ascorbate. Sixth one is PRIS, which is nothing but preservatives like sorbate, sulfate and nitrite, which are used to preserve foods. And the last symbol is CFU per ml, which is nothing but colony forming units per milliliter, especially competitive microflora, which are used in microbial fermentations. The safety and stability of foods can be ensured only by the way the hurdles are being used. The use of a single hurdle in higher amount range will cause serious effects to the quality of the product, deteriorating the product quality. Also, this leads to change in texture, color, flavor and overall acceptability of the product along with its nutritive value. So, in order to achieve the best product, the manufacturers should take care of consumer preferences and provide safe and stable products by the combination of different types of hurdles to give the consumers the best quality product, and also should prevent the entry of pathogens and microorganisms which spoil food. Depending on the types of factors which are specific to food products, the hurdles may be different for different types of food products, which are as follows. The initial load of the microorganisms present in the food. Secondly, the conditions which are favorable for the growth of microorganisms in the food product. And third one, shelf life or keeping quality of the product. Depending on the types of factors which are specific to food products, the hurdles may be different for different types of food products, which are as follows. The initial load of the microorganisms present in the food. Secondly, the conditions which are favorable for the growth of microorganisms in the food product. And third one, shelf life or keeping quality of the product. Now let us move on to the principles of hurdle technology. There are three principles of hurdle technology, namely homeostasis, metabolic exhaustion and stress reactions. Let us see each of these principles in detail. First principle is homeostasis. For the successful survival of the cells inside an organism, a uniform and stable ecosystem is required, which is called as homeostasis. The organisms should have a strong tendency to maintain their internal environment stable for the existence of a balanced homeostasis condition. The microorganisms in food will retain themselves in lag phase and will not multiply and will remain constant in their initial growth phase. If this stable condition is disturbed by preservative factors in foods, it is called Hurdle effect by making use of the hurdles which we are going to incorporate. Next is, to repair disturbed homeostasis condition, high level energy is required so that the repair mechanism can be prevented, which will lead to the synergistic effect of preservative factors. Synergistic effect is nothing but when you combine a single hurdle, there will be a particular result. When you combine 2 or 3 or more hurdles together, the effect will be better than by using a single hurdle. The energy restrictions may be given by anaerobic conditions like low water activity, low pH and low oxidation reduction potential. Now, the microorganisms should not overcome any of the hurdles present in the food product. By altering or disturbing the preserving factors or hurdles of the homeostasis mechanism, the multiplication of the microorganisms can be prevented, thus making the microorganisms inactive. Preservation of foods can be achieved by disturbing the homeostasis of the microorganisms. Now we'll move on to the second principle, which is nothing but metabolic exhaustion. Auto-sterilization in food products could be achieved by metabolic exhaustion, which leads to the death of germinated spores and thus ensuring the success of hurdle technology. There are a number of different kinds of bacteria, mold and yeast which overcome and sustain the minimum high temperature treatment. In a refrigerated storage, the hurdles used does not permit the growth of such microorganisms. So, their level has to be decreased rapidly in the products. Microorganisms in hurdle technology foods try every possible repair mechanism for their homeostasis. By doing this, they completely use up the energy and die. That leads to auto-sterilization of foods. One such example is preservation of foods by reducing the pH of the food sample. Then it is required to compensate the effect of protons by making use of weak acid preservative. Now let us see the third principle of hurdle technology, which is stress reactions. Due to the generation of shock proteins, some bacteria become more and more resistant or virulent under stress. The proteins are nothing but a family of proteins that are produced by cells in response to exposure to stressful conditions induced by heat, pH, water activity, ethanol, oxidative compounds, cold, ultraviolet light and starvation. These stress reactions increase the tolerance towards the stress which induce them. For example, sublethal heat treatment causes the microorganism to withstand high temperature treatment. Sometimes the stress reactions might have a non-specific effect, that is, due to a particular stress, the microorganisms become more tolerant to other stresses and they acquire a cross tolerance. The various responses of microorganisms under the stress might prevent food preservation and could turn out to be problematic for the application of hurdle technology. Likewise, the activation of genes for the synthesis of stress shock proteins help organisms to cope with stress situations will be more difficult if different stresses are received at the same time. Simultaneous exposure to different stresses will require energy consuming synthesis of several or more or at least much more protective stress shock proteins, which in turn may cause the microorganisms to become metabolically exhausted. Therefore, multi-target preservation of foods could be the key to avoid the synthesis of stress shock proteins, which otherwise could jeopardize the microbial stability and safety of hurdle technology in foods. Next, we'll move on to multi-target preservation in hurdle technology. If the types of hurdles in foods are in right combination, the homeostasis of microorganisms could be disturbed, resulting in a synergistic effect. Activation of stress shock proteins and repairing of homeostasis is much more difficult. For example, homeostasis can be easily disturbed by the addition of niacin, a biopreservative which damages the cell membrane along with lysozyme and citrate. Now let us move on to the example of multiple hurdle technology method. There are 6 examples which I will be explaining each one separately. Example number 1, as you see in figure, tells us about the food which has all the 6 types of hurdles like high temperature, low temperature, water activity, pH, oxidation reduction potential or redox potential and preservatives. All these 6 hurdles cannot be overcome by the microorganisms. So ultimately the food is safe for human consumption. Here is the first example where the intensity of all the hurdles is same, which occurs very rarely. Now let us see the second example, which tells us that there are different intensities given for different hurdles, which is the appropriate technology to be applied to different food products, and this also varies according to the types of food products. Now let us move on to the third example. Some of the hurdles alone here are sufficient for maintaining the stability of the product, since there is very less initial microbial load due to good hygienic practices or GHP. Let us go to the fourth example, which indicates that due to bad hygienic condition, which means that there is more growth of microorganisms initially, there is the presence of high microbial load in the product initially, and also here any combination of hurdles will not be sufficient to prevent the spoilage and poisoning of foods. And fifth example is for food with low initial load and rich in nutrients, which will aid the growth of microorganisms. So the hurdles for such a product has to be chosen in such a way the load of the microorganisms can be controlled completely. And the final example, which indicates the behavior of sublethally damaged organisms in food. If the bacterial spores are damaged sublethally by heat, the vegetative cells, and that is the disease causing cells derived from such spores, will lack vitality. Therefore, they will be inhibited by few or lower hurdles. Now let us see some of the limitations of hurdle technology. Synthesis of protective proteins due to stress of bacteria caused in them in hurdle technology results in preserving the foods. The stress reactions or cross tolerance may not even be present if we combine the different types of hurdles. There might be 3 possible results if the hurdles are given in different types of combinations. First type is additive effect. It explains the effects of individual components added to foods. Second one is the synergistic effect. It is an inhibitory action where one component depends on the other one for giving a beneficial or better effect rather than working separately. And third one is the antagonistic effect. It is the effect opposite to the other one, that is, where the mixture of the constituents required is higher than that of the individual constituents. The reason for using hurdle technology in food preservation is due to the combination of the first 2 effects rather than a single effect. Some studies show that combination of treatments are less effective in decreasing the microorganism level rather than the effect given by single treatments. This actually depends on the product type, hurdles used, and the intensity of the hurdles. The effects of combining hurdles were antagonistic. In some case, application of the hurdle concept for food preservation may inhibit the outgrowth but induce prolonged survival of microorganisms in food. The various responses of microorganisms under mild stress conditions of hurdle technology may prevent food preservation and can become problematic for the application of hurdle technology. However, no general statements can be made about the action of any particular preservative method on many other factors. Studies had synergistic effects observed in laboratory tests, and they were so weak to have no significance for practical food preservation methods. It is mainly for the commercial reasons that most of the combination products previously marketed in more quantity were preferred as straight preservations. Although Some combinations of hurdles showed less or negligible significant effectiveness for killing microorganisms in foods. Many promising hurdles have been identified for preservation of foods, but this application of the idea in the food industry has been largely restricted to the meat processing sector alone. Recently, emphasis is on a much wider potential application, for example, in bakery products, fish and dairy products. More specifically, the concept was introduced into mild processing, which is otherwise known as minimal processing of fruits and vegetables, where a very little processing is done to fruits and vegetables. For example, just cut fruits and vegetables can be done. However, there is only limited information available about the effect of combining different types of hurdles in these types of foods. The vast majority of preserved foods that are consumed in different countries depend on combinations of preservative factors for their stability and microbial safety. Therefore, it will be important to understand the interaction of different hurdles in various foods to find the types and the intensity of hurdles whichever are required or desired for controlling the microorganisms and to give stability to a particular food. The hurdle, which is supposed to have antimicrobial property, is specifically in Maillard reaction. Simultaneously, the flavor of the food products can be enhanced with the same concept, which is similar to curing of meat using nitrites. Some hurdles bring about unwanted results in food products which is not at all desirable. For example, chilling injury is one of the harmful effects which could occur in certain foods of plant origin by chilling of foods to very low temperature. For extending the shelf life of foods, optimizing chilling temperatures must be chosen. To inhibit pathogenic bacteria in fermented sausages, the pH of the product should be minimum. If the level of a particular hurdle in a food is too small, it should be strengthened. If it is detrimental to the food quality, it should be lowered, and an additional hurdle has to be combined with it. Another fundamental aspect of food preservation by hurdle technology is the synergistic effect of combined processes. This could be due to the disturbance of homeostasis within microorganisms. First, let us see about fruit industry. Initially, the fruits are washed to remove the debris, then they are peeled, sliced into smaller sizes, then blanched at appropriate or preset time and temperature, which controls the enzymatic activity. Then the fruit slices are soaked in sugar syrup and citric acid, which results in osmotic dehydration of the fruits, where the moisture content is removed from the fruits by the process called osmosis. Then the fruits are drained to remove the excess syrup present in it. They are then tray dried and then packed in appropriate packages to extend their shelf life. Along with these traditional methods, ultraviolet light, ultrasound and high hydrostatic pressure can be combined to develop novel fruit-based products. Next comes the meat industry. The hurdles used in meat industry include combining factors like lowering of water activity, acidity in the products and use of chemical preservatives like nitrite to stop microbial growth. The important hurdles of meat preservation include high temperature like heat treatment, low temperature like cooling and freezing, then water activity which can be achieved by drying by adding salt and sugar. Then acidity can be reduced by acidification. The redox potential or oxidation reduction potential can be done by decreasing oxygen or by introducing vacuum or by making use of ascorbates. Then comes the use of preservatives like sorbate and nitrites and many more. Finally, it is the competitive microflora. This is specifically for fermented food products. This is only applicable for non-heat treated products where no heat treatment is used, and preservation is done only with the help of microorganisms. Next comes the fish industry. Fish products can be preserved for long period by combining hurdles like salting and packaging. Here specifically modified atmospheric packaging or MAP is applied. This modified atmospheric packaging is a technique in which there is replacement of air in a package containing gaseous mixture like carbon dioxide, nitrogen and oxygen in a particular ratio according to the need of the products based on trials. But the composition of gas mixture is not controlled during the storage period. As the product respires, the gas composition changes accordingly. Next comes the dairy industry. Dairy products shelf life can be extended using hurdles like surface treatment and using vacuum packaging techniques for solid products like paneer or tofu. This paneer is otherwise known as cottage cheese. Along with these, mild heat treatments, acidification could also be combined. Finally, let us summarize few important aspects of hurdle technology. Hurdle technology is said to be an important fact for simple preservation of foods, as the hurdles in a stable product concertedly control microbial spoilage and food poisoning. It leaves the designed fermentation process unaffected. Because of their limitations, the individual hurdles may be set at lower intensities than it is required if only a single hurdle were used as preservation technique. The application of this concept has proven to be successful, as a proper hurdle combination results in achieving microbial stability and safety, and also stabilizes the sensory, nutritive and economical properties of a food.